Okay, as we discussed in a previous lecture, you can think of evolution as a two-step process. First, you have a random step, mutation of DNA, which is where we have generation of diversity in a population. Random changes, mostly caused by replication errors in DNA, causes us to have a population of genetically distinct individuals. Step number 2 is then natural selection. This is distinctly not random. We have differential survival and reproduction. Those individuals that have the most favorable genetic variants are those that are most likely to survive and get offspring. Now, this means that we can think of, and people did this early on, we can imagine two distinct fates for mutations. So a mutation that's detrimental, a mutation that for random reasons destroys a protein or destroys some aspects of an organism's fitness, will lead to what we call negative selection. That individual will have a smaller probability of surviving, and it might leave less offspring, it might leave no offspring at all. And this means that over time, this mutation will not be seen. Detrimental mutations, they pop up from time to time, but they cause the individuals to leave less offspring than their competitors. So over time, the frequency of those alleles will disappear, like we saw in the talk about models of growth and selection. The lower your fitness, if you have the lowest fitness, the frequency will decline. If, on the other hand, we have a beneficial mutation, a mutation that somehow enhances the function of a protein or makes some animal live better, making it more adapted to its environment, then we will instead have positive selection. This will have a higher fitness, and over time the frequency of that allele will increase until the point where it's fixed, meaning that these are the mutations that we see. So these were the ideas that people had early on about molecular evolution. It was that we now refer to as selectionist. It's a bit of a cartoon, but it sort of sums up the kind of thinking that was present at that time. The idea was that it was assumed that the majority of all mutations were detrimental. It's much easier to destroy a gene than it is to build a better version, and that these would therefore not be seen. They would be removed by negative selection. It was further assumed that the substitutions that we do observe when we do see mutations or changes in a population, that they are caused by adaptive mutations. That whenever some mutation is present, it's because it's good for something. Okay, some aspect of an organism is not just there for random reasons. That was the idea. Within this way of thinking, you could maybe divide the schools of thought into two groups. What we now call the classical school assumed that there would be a single predominant version of a gene, what they call the wild type, present in a population. That this would be the most fit, and that natural selection would therefore bring this particular allele to fixation. This would be the only allele present in a population. Whenever a new advantageous mutation arose, and that happens from time to time, then natural selection would very rapidly bring that to fixation, so this would now be the new wild type. Another school of thought said that, well, this is all fine, but if we actually look at animals, remember this was maybe in the 30s, before we had access to molecular methods for actually looking at the hereditary material. It was before we even knew the structure and what the hereditary material was. But in these days, just by looking at a population of animals, as we discussed earlier, you can see there's diversity. So they would say, okay, there must be genetic diversity also in population of animals. However, since they were also selectionists in this meaning, they would say, okay, there is diversity in the populations, but this polymorphism, all these distinct genotypes must be somehow be maintained actively by natural selection. There are ways in which natural selection can cause a population to maintain several different alleles. One example is that of sickle cell anemia. Sickle cell anemia is a point mutation in the globin molecule, which forms part of hemoglobin that binds oxygen in our red blood cells. This point mutation if you have one copy of this allele and one healthy copy of the gene, then you will survive. Some of your blood cells will get a strange shape when they are deprived of oxygen, and this, for various reasons, protects you against malaria. So this means that if you have one copy of this point mutation, you are protected against malaria. If you have two copies of the point mutation, you will die very young. If you have 0 copies of the point mutation, you're not protected against malaria. So in this case, there's a fitness benefit to being having one bad copy and one good copy. And this is one way in which natural selection can actually maintain polymorphism in a population. There are other ways also, but conceptually it's a possibility. So these were the ideas. Most mutations were detrimental. The mutations we see have some adaptive value and have been selected for. Those were the main ideas. However, later on in the 60s, when we first got access to electrophoresis, for instance, for analyzing polymorphism in proteins, it very rapidly became clear that polymorphism was much higher than anyone had anticipated, either in the classical or in the balanced school of selectivists here. Kimura and various others people, he was a Japanese population geneticist, then proposed what has now become known as the neutral theory of molecular evolution. The main idea in this was that in addition to the two classes of mutations we mentioned earlier, detrimental and beneficial mutations, there was a third class that was termed neutral mutations. Neutral mutations are mutations that essentially cause no fitness difference in an animal, meaning that there's no selection, positive or negative, for that particular allele. It simply doesn't make a difference in the function of a protein or in the fitness of the organism. And this means that whether or not these mutations will be seen over time is entirely stochastic. This is what we call genetic drift, a mutation that does not alter the fitness of an organism. It doesn't cause it to have more offspring or less offspring, or to be better at eating food or whatever other aspect of its life. Such a mutation, it's entirely random whether it stays in the population or not. And the proposal by Kimura and these other peoples was that the reason we have so much polymorphism is that a lot of this polymorphism is neutral. A lot of these mutations don't make a difference to the proteins. Now, it's important to understand that both of these schools, or both of these ideas about the role of mutations agreed on one thing, and I've tried to illustrate that with this pie chart. And that was that in blue here, the majority of all mutations are assumed to be detrimental, to be deleterious, and therefore to be selected against. Most of all mutations, everyone agrees, are deleterious. They destroy a protein. What they differ on is what the observed mutations are. So under the strict selectionist model, you would say that practically all of the observed mutations are advantageous. They're there for a reason. They're there because they make some protein better. And then maybe there's a small sliver of neutral mutations. The neutralist would say that the vast majority of observed mutations are probably neutral. They don't make a difference in fitness. And then there's a small section here with advantageous mutations. This means that in the selectionist view, most of the mutations we observe are there because they represent some sort of functional innovation. They have made some protein better. In the neutralist view, most of the mutations we see are probably there because they are present they make conservative changes to a protein. They alter some amino acid that doesn't alter the function of the protein, or they are changes in unimportant, maybe intergenic regions that don't play a role in regulation and similar, or in third codon positions that do not change the encoded amino acid and that won't have a big effect on fitness. So these two ideas about the role of mutation actually lead to quite different interpretations of diversity in an alignment of sequences, you can see. Now, one thing that everyone agrees on is, of course, that adaptations are caused by natural selection. Adaptations such as, for instance, the camouflage, that means that you are probably completely unable to see the gecko on the slide. It's hiding here. This sort of adaptation is caused by natural selection. Everyone agrees on that, whether you're a selectionist or a neutralist. They just disagree on what fraction of observed mutations are silent, are neutral like this. Other adaptations, such as for instance the beak shape of the Galapagos finches here, that cause each bird to have a beak that's particularly well suited to its preferred food. Everyone also, of course, agrees this is caused by natural selection. There's an advantageous mutation, causes the animal to be more adapted to its environment. It's selected for by the mechanism of natural selection. One observation that very much supported the idea of the presence of or the existence of neutral mutations was that of the molecular clock. It was discovered that if you observe some protein sequence in a whole range of organisms, comparing pairs of organisms that are further and further apart in evolutionary time, then there's a roughly linear relationship between the time span the two proteins have been separated and the number of substitutions you observe between them. So it appears that nucleotide substitutions accumulate roughly at a constant rate, and this has been called the molecular clock. Now, an observation like that is easily explained if most of the mutations we observe are in fact neutral and are not selected for or against, because then this rate will essentially be the rate of the appearance of neutral mutations.